what's important. Okay, I'm sorry for the delay. Hi, everybody who's here. If there are any problems with seeing me or hearing me, please just let me know. In the meantime, I'll just proceed. So today, I've set a live class for Monday because you have your test on Wednesday. But from next week on, we'll have a live class every Wednesday, unless you have a test. And in addition, I'm going to set aside not an office hour, but a half hour on a Thursday when you can actually contact me on Zoom to ask any particular questions that you have. So yeah, we'll see how that works. I wanted to have the live class today, especially because I want to go over the last part of the metabolism part, because I know how difficult it is for people to assimilate all of that material. The test on Wednesday will be 50 multiple choice questions. And in setting the multiple choice, I sit with the notes, with the PowerPoints, go through and make sure tests pertain to particular items. And I also use questions from your assignments. So use your assignments for revision. And I know that the last part, I think of the last assignment, will cover this part on metabolism that we did last Wednesday, and then what we're going to do today. I know that this is quite a difficult section, so I don't mind going through it slowly and repeating stuff. I'd rather teach you less and have you know it better than teach you more and have you learn it less well. Okay, so we've been talking about microbial metabolism. Actually, in actual fact, we're talking about metabolism in general, because most of the principles, most of the processes that we talk about in prokaryotes, we talk about prokaryotes, especially because this is microbiology. But most of the processes are very, very similar in eukaryotes. It's just that they take place in different parts of the cell. Eukaryote metabolism in many ways is almost identical to prokaryotes. There are a few differences, but really not a lot. Okay, so remember what we talked about. In fact, what we're watching happen inside the cell is the transformation of energy and of matter. And with any of the reactions that take place in the cell, we look at them and we can see, in most cases, biological molecules, the bonds that bond the elements of the molecule together, those bonds are established by putting in energy. That's either to build the molecule or to store energy if it's a fuel or whatever. When we break those bonds, we can extract energy. So the anabolic reactions in the reactions of building, those reactions tend to require an input of energy. The catabolic reactions, those with break molecules, tend to release energy. And that energy, in many cases, can be harvested. It can be transformed into a useful biological form. That's what we're watching happen. What we're seeing is that we're taking something which is a fuel. It's carbon bonded to, in this case, to lots of hydrogens. All of those bonds are high energy. They are high energy bonds. Think for a minute about where they came from. Let's take our molecule glucose. We use glucose as the simplest example, and we track it through all of these processes. Where did the energy in glucose come from? It's carbon, joined carbon to carbon, 6 carbons with attendant hydrogens attached to them. All of them covalent bonds. Those covalent bonds, the energy originally came from sunlight, because the glucose was originally made by plants. Its passage through all the things that eat the plants, et cetera, et cetera, et cetera. We end up, in all living things, we end up, glucose is a very good fuel, because locked into the bonds of the molecule, there is energy. And that energy is especially lodged in the electrons, especially the electrons of the hydrogens, and that is what we're watching happen. We're watching the cell break those bonds and harvest the energy, and it does it by shepherding electrons. And it shepherds electrons to processes where the electrons can donate their energy and get the cell to do something useful. What we want to watch is how we access a fuel like glucose, and we can now convert the chemical energy in those bonds. We're now going to convert that into energy which the cell can use in reactions. The cell usually uses energy in the form of ATP, adenosine triphosphate. So that is what we're watching. We're going to watch how we take glucose, here our starting material, and we're going to end up producing as much ATP from it as we possibly can. And we're going to follow all of those processes. Well, you've already been through a lot of them, so we can go through them relatively quickly. Let's just follow the diagram here. And I'll just take you, remember, I don't expect you to remember all of the intermediates. I'll tell you which ones I want you to remember. The first step of breaking down glucose, breaking glucose is called glycolysis. That means breaking glucose. So the first step in all of our process is this process called glycolysis. Here's glucose. Count the carbons. There are 6 of them. In order to break glucose is a very stable molecule. It's not that easy to split it apart. So we have to put in some energy at the very start. We're going to use up 2 molecules of ATP there and there. And what happens is we end up putting phosphate groups on the end of the glucose there. And that destabilizes the molecule. It's very easy to enzymatically break that 6-carbon sugar into 2 3-carbon compounds. And that is what happens. Never mind about this step. Just imagine that they both, because they get converted to this anyway. This, you can remember that, glyceraldehyde 3-phosphate. GP, it's sometimes abbreviated to. We call this the preparatory step because we have to expend ATP. What we want to do is harvest ATP essentially, but here we have to put in a little bit of energy to get the whole process started. That's a preparatory step. Now we go through a stage called the energy conserving stage, and watch what happens. We're going to we've got now 2 molecules of glyceraldehyde 3-phosphate. We broke this in 2, and we ended up with 2 molecules of glyceraldehyde 3-phosphate. So remember that when you go from here, what they're showing is 2 reactions all the way along. If I was just to follow 1 molecule, each of these would be 1, but we're following 2 molecules of glyceraldehyde 3-phosphate. So follow that through. Don't worry, again, don't worry about the names of the intermediates. You don't need to know that, but you do need to know what is happening as we go along, because there's some very important things. The very first step, look here, 2 NAD plus goes to 2 NADH. NAD is an electron carrier. It provides reduction potential to the cell. This is very, very important. These are electrons. We are stripping electrons out of those hydrogen bonds. We're stripping hydrogens, and we are putting them onto NAD plus, and we're ending up with NADH. When you look at this, when you look at NADH, what it's carrying is, in actual fact, is carrying 2 electrons, 2 hydrogen electrons, which we can use. We can use the energy from those eventually. This is reducing power. And this is what's really critically important in all of these processes. We run 2 NADs, we end up with 2 NADH. Now, look here. The next step, we actually make some ATP. We do it by stripping off a phosphate. So we make an ATP we make, for each glyceraldehyde 3-phosphate, we make 1 ATP, we make 2 ATP in total. Carry on down here, we produce some water, breathe out, you're breathing out water. That's one of the reasons why that is a waste product of this process. Here, we make another 2 molecules of ATP. So what have we ended up with? We had to put in 2 ATPs. We harvested 4, so we got 2 net, and we ended up making 2 molecules of NADH. Also, the end product of glycolysis is this, it's pyruvic acid. Remember, we've got 2 molecules of pyruvic acid. So let's now keep these all in mind, make a note of them, make a note of the products of that stage, because glycolysis can give you something. It gives you a small amount of ATP, and it gives you reducing power as well. It gives you active electrons that you can use. So each of those molecules of pyruvic acid is now going to be sent off. It's sent, first of all, this is called the intermediate step here. Pyruvic acid is converted to acetyl coenzyme A. Remember that, to acetyl coenzyme A. And in the process, we make another molecule of NADH. We've got another pair of electrons there that we can use. We produce some carbon dioxide. This is the first time we produce carbon dioxide. Breathe out. What are you breathing out? You're breathing out water from the previous step. You're breathing out carbon dioxide from this step. And you end up with this acetyl coenzyme A. Acetyl coenzyme A is sent now to this complicated cycle. A cycle is a metabolic pathway which continually regenerates its starting point. And in this case, it regenerates oxaloacetic acid. Oxaloacetic acid and acetyl coenzyme A together form citric acid. It's a 6-carbon compound. 1, 2, 3, 4, 5, 6. It's a 6-carbon compound. This now goes through this whole cycle. I don't need you to remember the intermediates, but all the enzymes. Remember, each step is governed by an enzyme. Huh? If you were doing biochemistry, they'd make you learn all of those. But we don't worry about it. Just remember, it starts with citric acid, ends with oxaloacetic acid. In the process, look what we make. Remember, this is 1 molecule of pyruvic acid. We're going to run the cycle twice. NADH there, that also makes some carbon dioxide. This more NADH, a little bit more carbon dioxide. Here it makes a small amount of ATP. Here it makes FADH2. FADH2 also carries a pair of hydrogen electrons. It's reducing power. It's an electron carrier. Here, another molecule of NADH, another pair of electrons for reducing power. And this step regenerates oxaloacetic acid. So now the oxaloacetic acid is ready to pick up another molecule and carry on and on and on and on. Make a note of these 1, 2, 3 molecules of NADH, 1 molecule of FADH2 per pyruvic acid molecule. So now the point is, we have to ask what is the importance of this NADH and FADH2? Well, here it is. In prokaryotes, each NADH is oxidized to produce 3 molecules of ATP. In eukaryotes, it's a little bit lower. You'll see the net amount of ATP produced at the end. It varies from 32 to 38. We usually accept classically 32 molecules as being the standard. So NADH can produce 1 molecule of NADH. 1 pair of electrons produces 3 molecules of ATP. FADH2 produces 2 molecules of ATP, slightly less. So this is what we do with our electrons. We're going to send these electrons off to this complex set of enzymes, proteins, and all sorts of things embedded in the membrane. In prokaryotes, these are embedded in the cell membrane, the plasma membrane around the cell. In eukaryotes, this process is going to take place in the mitochondrion. Like in eukaryotes, glycolysis takes place in the cytoplasm. Then this process takes place inside the mitochondrion. In prokaryotes, glycolysis takes place in the cytoplasm. This takes place at the cell membrane. It's the only membrane the prokaryote has, and this has to be embedded in the membrane. It relies on the membrane to act as an insulator. So first of all, just think, I think actually I've got a bigger one. Here's the membrane, and embedded in the membrane is this whole set of substances. They're all carrier molecules. Don't worry over much about the differences between the flavor proteins, cytochromes, and ubiquinones. Cytochromes you should remember because they are somewhat similar to hemoglobin. They have an iron molecule associated with them. They're somewhat similar to hemoglobin. There's a whole series of them. And what happens is that each of them can be oxidized and reduced, or rather reduced and then oxidized. So if we pump in a pair of electrons here at the start, we reduce the substance, and it hands those electrons onto the next substance. So in other words, it gets oxidized and it reduces the next. Same thing here. It gets oxidized and it reduces the next, on and on and on. As that happens, the energy level of those electrons begins to fall, and the energy can be harvested. As they go down this chain, the energy is harvested. And that is we'll see that it's harvested to produce ATP. I want you to only remember the broad details, the broad outlines of this, not necessarily the details. And what I want you to remember, first of all, look here. Here is a substance which can be reduced by electrons. This substance is reduced by NADH. NADH is oxidized. There it is.
Are you crazy?
I beg your pardon?
I'm so sorry, Professor. I accidentally unmuted it. I'm so sorry.
What's that?
Sorry. I accidentally unmuted my computer. I'm so sorry.
It's all right. It's fine. It's one of the things that happens. It's one of the things that happens on Zoom calls. At least you didn't swear terribly.
Wow.
Hello? There's somebody else is unmuted. Who is it? Please mute. Okay. All right. Okay. Let's start again. Here's NADH. NADH is reduced. There it's got a hydrogen and it's got 2 electrons. Comes down here. It reduces the substance. And when the substance is reduced, it in turn reduces the next substance. And it harvests a little bit of energy. As that reduction takes place, it harvests some energy from the electrons, and it uses that energy to pump protons. Protons are hydrogen ions. They are hydrogen atoms that have had their electrons stripped from them. And it pumps those out. This is the outside of the membrane. This is the cytoplasm. Those electrons, that pair of electrons is going to make its way through all of these substances. And there are another 2 here and here, which will also pump protons. They're going to pump protons from the cytoplasm to the outside. So now what do we end up with? Down here, we end up with a whole lot of protons on the outside and much fewer on the inside. That means we've got a gradient. That gradient is of 2 sorts. First of all, it's a gradient of charge. These are positively charged. There are more positive charges on this side than on that side. There is a gradient of charge. Second of all, it's a chemical gradient. This is an actual atom. There are more hydrogen atoms, even hydrogen ions on this side than there are hydrogen ions on that side. In other words, this side has become more acid. We've got a double gradient, electrical and chemical. We call it an electrochemical gradient. And we have a strong electrochemical gradient because this membrane is a barrier. It's an insulator. So these are going to accumulate on it. The drive is to equalize this, right? Equalize charge, equalize the amount of the chemical on either side. But this is a barrier. It's an insulator. And so what has got to happen is that somehow or other we're going to have to use these in order to do some work. What we've done is we've built up tremendous potential energy. This gradient here represents potential energy because these things want to move that way. So first of all, let's see how this is now harvested to produce ATP. It's harvested by this very complex enzyme here called ATP synthase. And ATP synthase, I think I've got here, you can see here, it's very, very complicated. And it's actually like a little motor. These proteins actually spin around and around and around. This is also a channel. It's a proton channel. And the protons actually flow through this. They're flowing through, driven by the difference in charge and by the chemical difference. They're driven by the electrochemical differential between the two. They flow through here, and when they do, they release energy, and that energy is harvested by this protein. Now, protein actually spins, and every turn, it makes one molecule of ATP. It's very cunning. And this is actually a very efficient way of manufacturing ATP. So let's just go back here. We've built up protons on this side. We build them up by running electron pairs through here. By the way, the FADH2 also contributes electrons, but it contributes one step down. So we don't harvest those with FADH2. We begin harvesting here. That's why FADH2 gives you less ATP than NADH. Look what we've done here. At this step here, we've regenerated NAD+, we've regenerated FAD+. That can now go back. It can go back to the Krebs cycle, it can go back to glycolysis and be reduced again. So what we're doing, we're oxidizing NADH, we're oxidizing FADH2, we're reducing here, then we're sending these electrons through a whole series of redox reactions embedded in the membrane. As they go, they lose energy, and we harvest that energy. We harvest it by pumping out protons to the other side of the membrane, building up potential energy, and we harvest that potential energy and we turn it into chemical energy. And that chemical energy is the bond between ADP and another phosphate to form ATP. Now, the end of the whole process is important because we can't just have electrons fly off into the cytoplasm. It doesn't work like that. The electrons, first of all, are being pulled through this gradient by the presence of oxygen at the end. Oxygen is extremely strongly electron negative. It is very attractive for electrons, and it pulls them through. The electrons then combine with oxygen and with protons. So if I take 2 electrons and I take 2 protons and I bind them to oxygen, I end up with H2O. I end up with water. Water is the end product here. Oxygen is the terminal electron acceptor. We have to have something that is going to sop up these electrons as they come through. And here it is oxygen, and then the protons are united to form water. And that's the whole process. So this is called oxidative phosphorylation. And it's reflection of the fact that oxygen is essential to the whole process. It is a phosphorylation because we're producing ATP from ADP plus a phosphate. And it's a very efficient, it's much more efficient than glycolysis. in producing ATP. And this is the process of cellular respiration. Okay, so as I told you, you'll see various figures for the amount of ATP produced, and you'll see anywhere from 32 to 38. Stick with 32. It's a good mean figure. It's actually mostly the figure which is calculated for eukaryotes. Prokaryotes, depending on how they work, they can have up to 38. And you can see here, have a look, and you'll see the glycolysis. There's really not that much ATP produced compared to the rest of the substances. And instead, by far the most, have a look here. This is the electron transport chain. We've got 22 compared to 6, 8, 10, 16 for all of the rest. I think that this is actually a better summary, I think. And this would be probably be for eukaryotes. Have a look here, glycolysis. We actually produce 4 ATPs. We produce 2 NADH, and in eukaryotes that produces 5 ATP. In prokaryotes it produces 6. Here, the intermediate step, 2 NADH as well, which will go off to produce 5 in eukaryotes, 6 in prokaryotes. And here the citric acid cycle, and then the electron transport chain produces 5, 17, 20. So we end up with a total of 32. The electron transport chain is an extremely efficient, combined with the Krebs cycle, is an extremely efficient way of producing ATP, much more efficient than just glycolysis on its own. And I mention that because there are, of course, many prokaryotes, especially, there are other things as well, but prokaryotes especially, which do not require oxygen. They are anaerobic. And they depend entirely upon glycolysis as their source of ATP. It means that their metabolism is much slower than those who use the aerobic respiration, but they can certainly manage, and many of them do. We'll talk about it more in a while, because there's some special strategies they have to adopt. There is a special case. The process that we've been talking about, we showed there free oxygen at the end. Well, that is the reason that we have to breathe. That's the reason we breathe oxygen. We require oxygen at the end of that electron transport chain as the terminal electron acceptor. And if free oxygen is not present, then essentially the process can't go on. But there are organisms which can use oxygen in another form. And that is, for the simplest example, are those which use the oxygen present in nitrate or in nitrite. You can see here, there's plenty of oxygen. It's just that it's not free oxygen. It's bound to nitrogen and nitrate or nitrite. And there are organisms which can break down nitrate to nitrite. There are other organisms which can break down nitrate to nitrite, and then break down nitrite to some other gaseous form like ammonia or else free nitrogen, and use even these. It does require an input of energy, so the yield is less, but still it allows them to continue to use that electron transport chain to produce ATP. And very typical of this is a bacterium you will surely encounter called Pseudomonas aeruginosa. It breaks down nitrate all the way to nitrite, and then nitrite all the way down to NO2. It's very efficient at doing this. We call this, has a strange name. It's called anaerobic respiration. It sounds like a tautology, but it's not. It's still using respiration. Respiration refers to that electron transport chain and the ATP enzyme at the end, ATPase at the end. But it is an anaerobic process in this case because there's no free oxygen for the cells to use. Okay, so there's a big problem with glycolysis, with using only glycolysis. The anaerobic bacteria can manage. They end up accumulating pyruvic acid, but their process stops right there. They can't proceed to fund the Krebs cycle, the citric acid cycle. They can't run an electron transport chain because that requires the presence of oxygen. Instead, they're stuck just with using glycolysis as a source of ATP, and they do produce they can produce a little ATP, and it's enough. If they perform glycolysis efficiently enough, they can manage. But there is a big problem, and it lies here. And that is, the cell absolutely requires reducing power. Even if the cell is not running the electron transport chain, reducing power is needed all over the cell for many different processes. And if, in fact, we run out of NAD+, this process itself will stop. We require for glycolysis to work, NAD+ is positively required. We have to be able to reduce NAD to NADH in order to produce the pyruvic acid and hence produce our little bit of ATP. So if there is not a source of NAD+, this process itself comes to a stop. So many cells will resort now to this process. It's called fermentation. And it is used to actually regenerate NAD+. That is its real aim. And they do it by taking the pyruvic acid and then by some means, it varies tremendously between different bacteria. By some means, they will actually reduce the pyruvic acid. Look here, we put it here as reducing power, NADH. We're reducing pyruvic acid and we're regenerating NAD+. That is so that we can use it here, so that this can carry on running. What method they use to do this varies, as I've told you, and what end product we end up with also varies a lot. This process of fermentation can produce various end products depending on what bacterium you're talking about. The commonest byproduct is lactic acid. So lactic acid fermentation, that is something which is absolutely familiar to us because we use this production of lactic acid in many, many food products. All of our cheeses, yogurt, all the fermented cabbage products, even fermented sausage, all of these things, they're all using bacteria which will take a sugar, which is actually lactose, the milk sugar very often, and they produce lactic acid. They reduce the pH of the substance that we're fermenting, the food. They reduce it so much that, in fact, other bacteria can't survive there. And we end up preserving the food with lactic acid. Sometimes they just produce lactic acid, that we call homolactic fermentation. Other times they produce a mixture of acids, that's very common, that we call heterolactic fermentation. There's another set of fermentations which are extremely useful for human beings, and those are alcohol fermentations. The most typical is not by a bacterium, it's by yeast, by brewer's yeast. And they produce ethanol, alcohol, and carbon dioxide. And these are both useful products. ethanol and carbon dioxide. Ethanol, that's why we ferment grapes for wine. It's why we ferment barley and malt, etc., for beer, and that produces ethanol. But this is useful in baking. In baking, we use the same yeasts. They also produce ethanol, but much more usefully, they produce lots of carbon dioxide. And the carbon dioxide is what rises the bread, allows the bread to rise, puffs up the dough with carbon dioxide. When you cook the bread, you bake off all the alcohol. And that's one of the reasons why bread smells so good. You're smelling the alcohol. But the bread rising is because of the carbon dioxide building up in the dough. Okay, so just remember, here we take in pyruvic acid, and we are reducing the pyruvic acid, and we're reducing it to some other end product. It depends what we're talking about. There are many, many end products, some of them very, very useful. So these bacteria, Streptococcus, Lactobacillus, Bacillus, these produce lactic acid. We use all of these, in production of things like cheese, yogurt, and sourdough bread. Saccharomyces, a yeast, it's not a prokaryote, produces ethanol and carbon dioxide. This one here, Propionibacterium, gets its name from the fact that it produces this acid, propionic acid. It also produces acetic acid. Clostridium produces a very foul-smelling set of products. Butyric acid, which smells like dirty socks. Butanol, acetone, all of these things. I'll come back to these in a second. Others produce, or we do have bacteria that produce ethanol, and I just want to point out something here. You see Escherichia coli, Salmonella, Enterobacter. Those are all very closely related. They belong to a group called the Enterobacteriaceae. They're all very, very closely related. And clinically, they are all very important, but they're difficult to tell apart. And so one of the ways that we can do that, one of the ways we can tell them apart, is to look at the end products of fermentation. There are some differences. For example, Enterobacter produces this substance, butanediol and acetone, and we can test for those in the lab. And if they're present, it's Enterobacter. If they are not present, then they're either Escherichia or Salmonella. It's an important way of distinguishing them. So remember, these end products are really useful. They're important clinically because it allows us to tell the difference. But they're also very important because look at these: propionic acid, acetic acid, butyric acid, butanol, acetone, isopropyl alcohol. These are all important solvents. And we use these bacteria industrially. We deliberately get them to ferment in huge quantities to produce these as products. So these fermentation products are very, very useful to human beings. Very, very valuable products.
Sorry,
Yes.
Professor, I have a question.
Go ahead.
So will all of these bacteria like always give a mixture of these products, or they can produce either or of this list?
You mean like these ones here?
Yeah, let's say, yeah, all the group.
Yes.
Is it always a mix of all of those?
It's a mix. Yes, it's usually a mix. Yeah. You can sway it by making sure that they have the right starting products. But yes, it's usually a mix. It's usually, you have to purify the stuff out of it at the end.
Okay, thank you.
Yeah. These ones just do this. So they're much easier to deal with. Okay. So again, this get clear in your minds the difference between glycolysis and fermentation. Okay, why fermentation is done in organisms which use glycolysis as their source of ATP. The other thing I want to say is we started our whole story by talking about glucose. We put in glucose. It's the simplest way of tracking what happens in all of these processes. But in fact, we don't have to use glucose. We can use many, many, many organic molecules as a fuel source. Some of them are used more easily than others. Our first recourse in a cell is to use carbohydrates. And in glucose, for example, or some polymer of glucose, starch in the plants, glycogen in the animals, etc., glycogen in bacteria as well. These are all carbohydrates. They're polymers of glucose, and they just would slot straight in to the chain of events that we've already described. But we can obviously, we have other things which build up, which can be used as fuel sources as well. The next in line, if for example, in us, if we run out of all of our carbohydrate stores, the next thing we'll start to use are lipids. And these are oils and fats, which are very, very stable, long-term storage products, which are there specifically to be used as fuel in hard times. When we've run out of everything else, we start to use the lipids and fats. They are not quite as efficient because they're still a very, very good source of energy, but they're not quite as efficient as a pure carbohydrate because they have to be slotted in elsewhere in this whole scheme. In fact, the lipids are broken down into fatty acids, which are pumped in down here just before the Krebs cycle. They go into the intermediate cycle there, step there. And lipids, fats, don't forget, they have a glycerol molecule, very similar to carbohydrates. It's actually an alcohol. And that gets transformed and plugged in there. You don't need to remember the details. All I want you to realize is we can use lipids, for example, as a fuel source, but they don't go through the whole process. They get plugged in later and their yield is thus a little bit lower. If you are absolutely starved, if you have no other source of nutrient at all, you've run out of all your carbohydrates, you've run out of all your fats and lipids, then you can start, then your body will start to use proteins. This is a very dangerous situation, but it is possible. Proteins can get broken down to their amino acids, and depending on what the amino acid is, which metabolic pathway it goes through, they can be plugged in at different points in that chain of events, and they can be used. This is a very dangerous situation. This is the sort of thing we see in children who are extremely, extremely malnourished. And the problem is that some of those proteins are essential proteins. For example, they may start to use neurological proteins. They may start to use proteins of the brain and things like that, and end up really doing serious permanent damage once you get to this stage. Just remember, the various organic molecules can be used as a fuel source, not only carb. We don't have to depend on carbohydrates. Okay, and I just want to talk a little bit about photosynthesis. I'm not going to go into a huge amount of detail about it. There's a couple of things to bring to your attention about photosynthesis. In actual fact, photosynthesis is almost the exact opposite of respiration, of cellular respiration. What did we do in cellular respiration? We took glucose molecule, C6H12O6, and we broke it down. We burned it in a series of very, very careful enzymatically controlled steps, and we harvested the energy from the bonds that made the glucose, and we ended up with the simple molecules, carbon dioxide and water. Carbon dioxide and water we breathe out, and they are lost as waste products. So that's cellular respiration. What is photosynthesis? Photosynthesis, we take carbon dioxide, we take water, we take energy, and we bond carbon dioxide and water together, and what do we make? Glucose, C6H12O6, from CO2 and H2O. The bonds, the energy in the bonds that build that glucose molecule, that is light energy. And that is the miracle of plants, is that they can take light energy and they can actually, in the end, convert that light energy into chemical energy, which they put into an organic molecule. Once that organic molecule is made, the plant uses it for sure, but other things can use the plant, and it can use the plant molecules as a source both of carbon and of energy as well. Now, the whole process of photosynthesis is very complex, and in energetic terms, it's actually very expensive. It requires a lot of metabolic energy to be put in, but it doesn't matter because the energy is free. As long as the sun is shining, the energy is free to the plant. So we'll see that there are two parts to the process of photosynthesis. The first is called the light-dependent reactions. And these are the reactions where we convert light energy to chemical energy. And we do so by creating ATP, the old friend. And it's called NADPH. It's very similar to NADH. This is a plant product predominantly, NADPH. So we create some ATP, but look what is this? This is reducing power. We've taken the light energy and we've energized hydrogen electrons and we've bound them here. This is reducing power, and we're going to use that reducing power. There is another set of, think of what we said about photosynthesis. We end up producing a sugar at the end, an organic molecule. We think of this glucose. This is the simplest example. The sugar, we first of all converted the sun's energy into chemical energy, these two compounds. Now we're going to take carbon and we're going to take water, and we're going to we're going to rearrange the atoms of carbon dioxide, and water. We're going to rearrange the atoms to form a sugar. When we bind the carbon dioxide into a molecule like that, we call it carbon fixation. And this is an independent step. It's got some very confusing names. It's the light independent step because it doesn't require light to drive it. It used to be called the dark reaction. That is a very bad name for it because it doesn't have to be in the dark for it to take place. It's just that it doesn't need light to take place. And what happens in these reactions, the light independent reactions, ATP and NADPH are used as a source of energy. And they're used as a source of energy to bind carbon dioxide and water together to form a sugar. They do so in a very complicated cycle called the Calvin-Benson cycle that you'll see in a minute. I don't need you to remember all the details of it. What I do need you to remember is photosynthesis has these two parts. There is a light-dependent part where we convert sun's energy into chemical energy, and there is a light-independent part where we use this energy to make sugar. I'll just show you very quickly the bare bones of the light reaction. I'll show it to you in two forms. I've first of all explained to you this. You see here these two molecules here of chlorophyll. Chlorophyll is a very special substance. It's a pigment. It absorbs light. And when it absorbs light, some of its electrons become enormously excited. They start to fly far away from the proton. And they, in fact, leave the chlorophyll, and they can be passed through an electron transport chain, just like we saw before. We actually do it in pairs. A pair of electrons rushes through a set of redox reactions, just like we saw before. And in the process, ACP is made. That's really rather similar to cellular respiration in a way. Okay, so they're rushing through here and forming ATP. They lose energy, and they are absorbed by this chlorophyll, which is now hit by light itself. Here's another light beam. It's hit by light and loses 2 of its electrons. So it gains 2 electrons, it gets reduced, but it loses 2 electrons, which it again contributes to an electron transport chain. And this time it makes NADPH. Those are our 2 products of the light reaction, ATP, NADPH. There is something to notice though. We've sent off electrons, right? We stripped electrons from here. We ionized the chlorophyll. It can't remain like that. It has to gain electrons from somewhere. Otherwise, this whole process is going to stop. It can't carry on and on donating electrons. It's got to have its electrons replaced. And this is the really amazing part about, one of the most amazing things about photosynthesis. Photosynthesis actually breaks water. It breaks water. It takes a water molecule and it strips off electrons from the water molecule. There's this H2, strips off 2 electrons to give it back to the chlorophyll. In the process, it produces protons. Guess where those protons are going to be used? They're going to be used in the production of ATP and NADPH. Okay, those much the same as we saw. It's going to involve electron pumping, proton pumping, and all the rest of it. In the process, if I strip off the hydrogens off water, what I end up with free oxygen. And that is why plants produce free oxygen as a product of photosynthesis. They produce, first of all, glucose, C6H12O6, and they produce free oxygen. That is the oxygen we breathe. Okay, now just think of our previous formula, right? What did we say respiration was? Glucose gets broken down to carbon dioxide and water, and we harvest the energy. Photosynthesis, we take carbon dioxide and water, we bind them together with the sun, with energy, with sun's energy, which we convert to chemical energy, and we release oxygen. Photosynthesis, the formula for photosynthesis is essentially the inverse of respiration. And this is where our oxygen comes from. And it's going to be where our source of energy and carbon come from as well. Here they are. You don't need to pay much attention. This is really just to show you how very similar in many ways, this membrane-bound system of electron transport is. And here we hit light. There goes the chlorophyll electrons. Ding, 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 ding. Here's light again. Ding, 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 ding. We end up making ATP, also by ATP synthase, same as before. And we end up making NADPH. This is really just to show you that it's membrane bound and that we're pumping protons just as before. We're using essentially a similar kind of system. That gives us some ATP and it gives us reducing power. And we now, I think I've got a better one than that. So those are the light dependent because they're driven by light. This is the light independent step. This is called the Kelvin-Benson cycle. It was actually discovered by these 2 different workers, Kelvin and Benson. And the Kelvin-Benson cycle is what actually ends up producing glucose or a sugar of some sort. It can be various sugars. You'll see here it says glucose and other sugars, but we will think of just as being glucose for the moment. So what happens in the light independent step? Well, we've got to fix carbon dioxide. Here it is here. We're going to take molecules of the gas, carbon dioxide, not an organic substance at all. It's an inorganic substance, carbon dioxide. We're going to fix it. The way we do that is we use this substance here. It's called ribulose bisphosphate. There is a very important enzyme here called Rubisco. And Rubisco is the enzyme which governs this step. And rubisco enables us to bind carbon dioxide to ribulose bisphosphate. We form a large, very unstable molecule which breaks up into these molecules of, it's called phosphoglyceric acid. Now look what happens as we go around. All I want you to notice, you don't need to worry about the steps or how many, etc., etc., etc. But all I want you to notice here, look what we're doing. We're taking ATP and we're producing ADP. Where did the energy go? The energy is going into this here. Here, we're going to use up masses of NADPH. Here it goes here. We're going to use up masses of NADPH. We're forming substances and deliberately enzymatically destabilizing them, and they will eventually crack and form glucose. What we have to do is what we want to do, we want to form a 6-carbon sugar. Carbon dioxide's only got 3 carbons. And we have to regenerate this here, ribulose bisphosphate. So what happens is this here has to swing around 6 times. You have to swing around 6 times for each molecule of carbon dioxide. You have to swing around 6 times to produce 1 molecule of glucose. So we're going to, this is what it says, 6 here, 6 ATPs, 6 NADPHs, 3 ATPs here to regenerate this. It's very, very, very expensive. It's a very expensive process, but it doesn't matter because this is free. You've got, as long as the sun is shining and you're driving your light-dependent reactions, you've got as much ATP and as much NADPH as you want. And you can drive this around and around and around and around and around and produce masses of glucose as long as you have plenty of carbon dioxide, as long as you've got plenty of water. You can bind them together using the sun's energy and end up with a good organic molecule, glucose. Sometimes plants do produce other sugars as well. That's all I need you to know about photosynthesis, okay? Just the bare bones. Remember, this is a step to fix carbon dioxide, and it swings 6 times around to produce 1 molecule of glucose, and in the process, it produces that to continue the cycle, ribulose bisphosphate. Okay, so let me just look at the time. Let's just give it another couple of minutes. I'll just do this slide, and then we can move on to some questions. I want to just impress on you something about the prokaryotes, and that is, you will already have gathered that there's tremendous metabolic diversity in the prokaryotes. All of the metabolic processes that we see in our own cells or in plants or whatever, these all evolved in the bacteria. They are an incredibly metabolically diverse group. And there are many, many ways that bacteria actually obtain energy and their carbon. Those are the 2 things which all living things need. We need energy, we need an energy source, and we need a carbon source. We need the molecules to build our biomolecules. We need the energy for the bonds in those biomolecules. So all organisms, that's what we need. We're all going to need carbon source and energy. So if we look at the organisms, we find we can divide them immediately into 2. Those which use light as a source of energy, and those which use a chemical as a source of energy. By a chemical, what do I mean? Well, we use chemical. Our food is a chemical. We're using our food as a carbon source and as an energy source. Plants, on the other hand, things like that, they use light as a source of energy, but they still need a carbon source. So what we've just talked about, the system of photosynthesis that I've just talked about to you, that used carbon dioxide as a carbon source. Here it is here, carbon. We can stop here. Don't worry about this part down here. Okay, you can just stop here and make a note of this. The photoautotrophs use carbon dioxide as a carbon source, and they use light as a source of energy. These are all the things like plants, the photosynthetic bacteria, all of those sorts of things. That is what they do. They are photoautotrophs. There is another group, however, of bacteria which are very strange, and they use light as a source of energy, but they use organic compounds as a source of carbon. They're very peculiar. They are called the photoheterotrophs. You are not going to encounter them at all. You're not going to ever talk about them again. Just be aware that they exist. Very important in sediments, by the way. So they have an important ecological role to play, the photoheterotrophs. So those are things that use light. What about the things which use chemicals? Well, that is us. We use chemicals. And anything that uses a chemical as a source of energy is going to be called a chemotroph. Chemotroph feeds on chemicals. It's a chemotroph. But there are organisms which use a chemical source of energy, but use carbon dioxide as a source of carbon. Anything that uses carbon dioxide as a source of carbon, we call an autotroph. These are photoautotrophs because they use carbon dioxide as a source of carbon and light as a source of energy. These are chemoautotrophs. They're chemoautotrophs because they use carbon as a source of carbon. I mean, carbon dioxide as a source of carbon, and they use chemicals as a source of energy. Those chemicals that they use, it depends very much on what they are. There are many, many, many of these organisms, which we call the chemoautotrophs. They use the energy in ordinary inorganic chemical bonds as a source of energy, and they use it to fix carbon dioxide and use the carbon dioxide as their source of energy, of carbon. Many, many, many of them. Some of them use hydrogen sulfide. Some of them use iron. Some of them use nitrates, ammonia, things like this. There are many, many, many of them. They're very diverse, a very, very diverse group. Also not clinically important, but very, very important ecologically. So if they're not using carbon dioxide, if you're not an autotroph using carbon dioxide, if you have to be using other compounds, organic compounds, as your source of carbon, just as we do. And those we call the chemoheterotrophs. And the chemoheterotrophs are divided into two. Don't worry any more about than this. There are some which do not use oxygen as the final electron acceptor. They don't use free oxygen as the final electron acceptor. Don't worry about it, okay? Just think here, this is us. We use oxygen as our final electron acceptor. We use organic compounds as our source of carbon, and we use organic compounds as our source of energy as well. We are chemoheterotrophs, and we share that with many, 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 many bacteria as well. Okay, I'm going to stop right there, and I'm just going to ask you guys if you have any questions about this or about stuff that you want to talk about for your test on Wednesday.
Really quick question, Professor, for the test. Can you hear me?
Yes. Yes, I hear.
Okay. So would you say that we should reference back on the slides, but also on the questions, on the assignment, like the 78 questions, I believe? Yes. Do that. What would you say majorly we should focus on? You say the questions?
Well, it's hard to say because some of the questions are going to cross over between assignment and, but what I want to tell you is I'm not going to dive into your textbook and ask you stuff that has not been covered either in the assignment or in the classes.
Got it.
Okay. So, and I'm not looking for obscure factoids.
Got it.
You'll be able to take afterwards. You'll be able to look at the test and go to your slides and go and say, yes, it was that slide that it was referring.
Okay, perfect.
Yeah.
Thank you.
Sure.
So basically, you mean that the slide and the homework, right?
Correct. Yes.
Okay, thank you.
You go through the slides and go carefully through the homework. Even though I must say the assignment also does sometimes take you a little bit broader than we cover in the class, but nonetheless, I'm using the assignment as a source of question, although I may reword them. But if you've done the assignment, then you'll recognize what I'm asking you. As far as the assignments are concerned as well, some people seem to think that the assignments are almost like a test, all right? But they're not. You are free to work through the assignment just as you like. You can use whatever source of information you like to answer those questions. That's the whole point. It's to get you reading and checking back and looking at your slides, looking at the textbook, etc. And you just work through them as well as you can until you end up with what you think is the right answer. How you arrive at that answer is your business. The whole idea is to get you to work through the stuff on your own.
I just have a question. So like some of the homeworks, like let's say I know that like some of them have like 5 answers, so like I'll get confused and like I'll answer like one of them that's almost the same. Like I know there was one of them with like spelling with an uppercase S and a lowercase S that I answered correct. So when I went back into the homework, it graded me wrong for the question. If I like redo it, does it change the grade of the
if you haven't submitted, you can redo if you haven't submitted. Yeah. Once you submit, I can, if you really want me to, I can undo the assignment, but I can't undo an individual question.
Okay,
I can just open the assignment again for you.
It'll change the grade, right?
Yeah.
Okay,
yeah.
perfect. Thank you.
Yeah. But most people seem to be doing extremely well on the assignments. Most people are doing getting really high marks, which is what I want.
Is the exam covering chapter 5?
I beg your pardon, is there a what?
The exam on Wednesday, is it covering chapter 5 too?
Yes.
So everything we covered until this class, right?
Correct, yes, correct.
Okay.
Yeah.
I'm sorry, Professor, do we have to have a lockdown browser or any such thing?
No,
for the...
no, I'm not using lockdown. I'm
Okay.
not using lockdown at all. I'm trusting you to be honest, and also I must just tell you the timing is extremely tight, and anybody who's trying to look up answers, you're going to fall behind very, very quickly. You must be able to answer these questions on your own. The other advice I always give to all of my classes, as far as multiple choice is concerned, people make a terrible mistake with multiple choice, and that is they look at the answers first before they read the question properly. And people really do. You should blot out the answer list from your mind. Look at the question, read the question carefully, and answer it in your head before you look at the answers. Get the answer in your head, and then look for the most similar answer in the list, in the multiple choice list. And that'll be it. Doing it any other way, it can really end up confusing you. You may know the answer, but then you start reading all the answers and they just confuse you. You lose your confidence. If you answer it confidently in your mind before you look at the list, you'll be able to spot the answer right away and with confidence know that that's the answer. That's my advice anyway.
Professor,
Anyone else?
I have a,
Yes. Yeah, go ahead.
how much time do we have for the test?
An hour.
An hour, and it's 50 questions, correct?
50 questions, yes.
Awesome. Okay, thank you so much.
Uh-huh. Anyone else?
And will the test be in like the assignment tab?
It'll be in quizzes. You just go to quizzes and you'll see it there with it'll say microbiology test 1.
Okay, thanks.
Yeah. And you have it. You should log in at the class at the correct class time, 4:05, and then you have an hour. Once you logged in, it's a few minutes, give or take. But from the time you log in, you will have 1 hour to answer. All right. If nobody else has anything else, then let's end off. Thanks all for attending. And we will repeat this in next week. We'll have a live class on Wednesday, not on Monday, but on Wednesday. And anything else, I'll be in touch with you. All right?
Thank you,
very much.
Professor.
From
Yes,
now on, we
pleasure.
will always have Wednesdays online.
Yes.
Okay,
Yes.
thank you.
I'll do a live class, but the class is recorded. It's recorded and posted just the same as all the others. Because we have people, who are not able to attend at the class time. I mean, that's the whole point of doing online classes. So, all right then, everybody. If there's nothing.